Hello and welcome to Channels for Nextflow podcast. You are listening to episode 44, and today we're really lucky to have Alex Pelzer. He's a key figure in the Nextflow and NF Core community. Many of you, I'm sure, will know him and have heard of his name before. And I've managed to twist his arm and bring him onto the podcast to talk to us all about the exciting topic of regulation around bioinformatics and Nextflow pipelines. Alex, thanks very much for joining me today.
Thank you, Phil, for the invitation.
Before we get too stuck in, I want to do an early flag for our upcoming Nextflow Summit. It's in Barcelona at the end of October. You can find out more details at summit.nextflow.io. We have an early bird discount. If you go to register, you can see all the details. 20% off on registration fees, and that expires at the end of August. So you don't have many days left. So I just wanted to flag that to anyone who's listening, that it's going to be a really good event. We've got training, we've got NF Core Hackathon, and we've got some fantastic speakers. One of whom, of course, Alex. You're going to be giving a talk. So yeah, if you're listening to the podcast and you can't get enough of Alex's voice, you know where to find him. Are you going to be at the hackathon as well?
I plan to go for the full week, so with Summit and the hackathon, and we were already thinking about doing something on the hackathon, specifically on regulatory as well.
Fantastic. So if this is your thing, if you're interested in working around this, then absolutely valuable time to come and meet other people interested in it and get up to speed.
Absolutely.
Alex, tell us, you and I, we know each other, longer than I like to think now. Probably coming off 7-8 years or something now. I think maybe we met at the first Nextflow meeting ever in Barcelona in 2017 or something.
Yeah, late 2017 it was.
Both of us were working in academia at the time. I was at SciLife Lab in Sweden and you were at QBiC and tubing in Germany. Tell us a little bit about your background, how you got into using Nextflow and what brought you here today.
Yeah, I was working on bioinformatics pipelines like pretty much everybody in bioinformatics, I think, but without using frameworks like Nextflow or Snakemake or others. So in the past, I was using my own more or less self-developed Java-based framework, which had also concept like modules and things like that, but was exclusively only used for ancient DNA analysis. So the only pipeline that I was able to write was one of my PhD projects, which was called Eager, which some of you might know because it's already on NF Core as well, but now in Nextflow, obviously, and there's also some other folks now working on this because I actually moved away from ancient DNA research.
I feel like writing your own bioinformatics pipeline software was like a rite of passage for anyone working in the field 10 years ago. You're another one to add to that club.
Yeah, pretty much. Yeah. Some people say it's also about writing your own aligner at some point in your career, but I think it also boils down to writing your own bioinformatics workflow. Yeah, so I started working with that. And then when I was about to finish my PhD, I was looking for options, actually, where to work, what do I want to do next. And a good friend of mine who was also very much involved in the early days, so Sven Fillinger, was asking me whether I would to join a QBiC, because they were also having interest in setting up standard core unit bioinformatics pipelines for daily analysis. And back then I thought, okay, yeah, that's a cool thing to do, but I need to come up with a good solution on how to do this properly. I was actually giving a talk about the Eager pipeline in 2014, 15-ish at the HPC conference in Frankfurt, where I met some people who were early involved in Nextflow as well, but I didn't look into it further back then because it had already developed that framework for ancient DNA analysis. I couldn't really squeeze this in anymore at that point. But at that point, I thought, hey, it might make sense to actually join one of these Nextflow meetings back then in Barcelona. At the CRG back then was pretty small. It's not too many people, actually. 30 people, maybe, I don't know.
Yeah, like 30 people. It was like a small meeting room, wasn't it?
Yeah, it was really small, but also quite nice because it was easy to talk to people. And that's the way we met, I think, the first time in person.
Yeah. If you look at all the people that were sat in that room in 2017, it's amazing how many of them have gone on to become friends, colleagues, how many of them are still in the Nextflow space. It was a real formative group of people, I think.
Yes, absolutely. It was really nice actually to talk to people, to be able to actually also influence, to also collaborate on things. And I think I talked about it with you because NGI back then had a lot of pipelines already out. We had already a little bit of a standardization effort. And we thought, okay, let's not just reinvent the wheel, let's just collaborate. And I think that's when it kicked off, right?
So 3 podcasts ago, episode 41 or something, was a podcast I did at SciLife Lab with Fran and with Maxime. And we were talking a little bit about this history of NF Core. It's nice to chat to you now and see the other side of that coin. I remember you getting in touch. I think it was about a single cell analysis pipeline you wanted to build using the NGI framework and stuff.
Yeah, so that was back then quite nice. And I thought about using NGI pipelines in the beginning, and then you were bringing it up to rebrand them, to put everything under the NF core hood. Then others also chimed in. There was also Andreas Wim from A-Star Institute in Singapore, so who wanted to contribute. And there was already a couple of people involved very early on, bringing in their own expertise, bringing in their own pipelines. And I think from then it became history. People joined in, started collaborating, started developing, started contributing. I think that was quite nice to see as well for me in the new job at QBiC, because all the internal developments then also were more or less streamlined into contributing directly to NF Core rather than developing in-house stuff anymore. I think that helped a lot later as well.
So you co-founded NF Core with me, and we set that up, and you converted everyone at QBiC to using NF Core, which is a legacy which lasts to this day.
I think so, yes. So my knowledge, at least, there's still lots of people at QBiC doing NF Core work, doing contributions, using pipelines. It's not a question anymore whether you are using NF Core pipelines as the standard, the default. Back when I started, that wasn't the case. So that's a difference now. It's been like this everywhere nowadays, that people are switching from in-house developments mostly to the public NF Core pipelines.
So you finished at QBiC and moved on into Pharma.
Yeah, after a couple of years at QBiC and building NF Core there and also contributing to project work there, I decided to move on towards something different. Towards my case, this was a move to Boehringer Ingelheim, to a pharma company. At Boehringer Ingelheim, we also use Nextflow quite a lot. Actually, all the in-house pipelines are Nextflow as well, or have been migrated to Nextflow. So, yeah.
I was going to say, you joined QBiC and you converted QBiC to be a Nextflow and of course shop. Was that your plan with BI? What was it like when you first joined? Were there many people using Nextflow already?
Yeah, a good friend of mine called me an NF Core terraformer sometimes. Wherever I enter, I seem to convert people to using NF Core tooling and Nextflow tooling. But it's a bit difficult because QBiC is rather small. We're talking about like 20, 30 people. Like when I joined QBiC with BI, that's a different story. We're talking about hundreds of people in bioinformatics. It's more difficult. Also, there's different applications. It's not always the same shoe does not fit all of them, obviously. However, I also see that the uptake is actually speeding up, and people are now also more happy with how to use Nextflow pipelines than they were in the past. So I think it's been moving that way. Yeah.
And anyone who's listening who's not familiar with pharmaceutical companies, and can you tell us a little bit about what BI is and what BI does?
So we're developing medicines for oncology, inflammation, cardiometabolic diseases, and are trying to do clinical trials. So we do both the discovery part, early clinical research part, as well as then also developing stuff for application in humans in the end. Also animal, we also have a part of the company doing animal health. So what we're doing is applying these pipelines also on biomarker analyses across projects, across clinical trials, and trying to figure out whether we see certain trends in the data that could, for example, explain the mode of action of a medication, for example.
And is this mostly genomics data, like DNA sequencing, RNA sequencing, or a bit of everything?
I think it's a bit of everything, but the focus is strictly on genomics. Transcriptomics is the standard thing nowadays, but there's also other applications. There's also a bit of proteomics, these panel-based approaches where you have a couple of hundred targets and we measure them because it's easier in clinical settings to measure these in a more standardized way. Same for RNA-seq, for example. It's exploratory analysis in 99% of the cases, but in the end, it's also helping you to confirm whether there's some trend in your data that you also can see, for example, on clinical safety or efficacy of data or something.
The title of this podcast is Regulatory Revolution. So with that context of the work you're doing, what does regulatory mean to you?
I think it's a bit of a catch-them-all phrase, honestly speaking. When we had the kickoff with the special interest group around regulatory, we had lots of people joining, lots of people with interest, but they came all from a bit of a different background, a bit of a different angle to this. And for us here in the pharma setting, it's a bit of a different angle than I would envision is for, let's say, a hospital where they do clinical diagnostic testing versus what we do in clinical trials, for example. For me personally, and I would say regulatory is a framework that is set by authorities that you have to obey, that you have to make sure that you do certain analysis in a way that they are reproducible, that you can guarantee that what you did is actually making sense, that you can guarantee that the quality of your analysis actually matches certain criteria, certain standards, meaning for every sample at a hospital should be already in a very good shape. up to pipelines where we do their data analysis and then also downstream analysis. And we should actually be able, upon request, to actually tell them and show them exactly, this is how I did it. This is the pipeline version I use. This is how I did the testing, that this pipeline actually does what it should do, that it's really fit for purpose for this type of analysis. And then also downstream, that you can really fully explain what's going on here, what is done and how it is done and who reviewed this. So there's multiple things involved there in the entire chain that you should actually be aware of. And that's, for me, that's the part for regulatory, if you look at it from a pharma context, obviously.
And what are the consequences of this? If a regulatory body checks and doesn't think you're doing enough or not doing the right thing, what happens?
There are regular checks, and I think they're there for a very good reason. If you don't do 100%, and then they will mark these as findings and will tell you, okay, you have to improve here. It depends a little bit on how severe these findings are. If they are severe, it's getting to a point where there will be consequences for you as a company, which also will mean potentially a lot of money. In the end, what you would like to do is to prepare for these types of audits. You would like to be able to show people in authorities, we're doing the right thing, we're doing it properly, we're doing it fully fleshed out into all of the details that you need to be aware of, so that anybody out there could, given the data, then you can point them to, this is how we did it. If they can reproduce it, then they will have some trust that this has been done properly.
So what's the bulk of the day-to-day work for this? It sounds like there's a lot of record keeping, you keeping notes of what it is that you've done. Is there other types of work you need to do in terms of looking ahead of time, choosing pipelines or written documentation?
Yes, so it boils down to do a lot of record keeping, honestly. If you do analysis, you have to do a lot of record keeping. You have to make sure that you know exactly what you've done in the past. There's a lot of quality-driven documentation on how these steps should be done, ideally. Then there's also scouting. We know clinical trials usually don't run within 6 months or something like that. They run on a much, much longer time scale. So if you're running these across a couple of years maybe, or some of them might just start in 2 years and run over 3-4 years, you also have to scout for new methods. You also have to scout for new analysis technology. So we usually get involved quite early in this development. So whenever there is a new trial planned, they select assays, and then we give input on that from a bioinformatics perspective and check, okay, is the assay even doing what we expect it to do? How do the readouts look like? Can we have some test data? Can we run some pipelines on that already? In many cases, this is standard stuff, so like RNA-seq, for example. I mean, it's not like you reinvent the wheel every six trials or something like that, but in other cases, it might be something where it's much more difficult because it's something not as established. B-cell receptor sequencing, for example, is a topic that is currently en vogue a little bit. So that could be something of interest as well.
And presumably in some cases, there is no existing analysis method and you have to go and write something.
Very rare cases, although I would say that's a bit more of a problem for the early clinical people, because usually they're the ones who are working with this type of data way, way earlier than we have in the clinical setting. So in many cases, we also get some notification from colleagues in-house already. This is what we've done in the past. Maybe you want to check it out. And then we can either take the same tooling or figure out, okay, there's a more production-ready way to do this for a clinical trial that we would like to validate, that we would like to check whether it's fit for purpose.
And so if someone comes to you and it's a new genomics method that you haven't done before, you do some research to see what's available already, what things are you looking for and what work do you typically need to do once you select an existing tool?
So in my case, I usually tend to acquire some test data if this is possible. So if I can get test data from some public, open source, academic background, that's usually a first step. So applicability is for me something that I really like to focus on. Some people just publish stuff and they're there for publishing. And if you look at the tool and the coding in the end, you figure out bugs and issues with it. It's a bit of a different story if it's open source stuff, then usually you can contribute to. So there's a decision that you have to make at some point. Do we invest into this? Do we actually make the tooling work for us? Do we invest at least as much that it works for us for our use cases? Or do we reinvent something completely from scratch? In many cases, it's enough to invest a little bit to make it work for your use cases, and that's what we intend to do. What I also look for is that it's usually open tooling if possible. So that it's open source ideally, that it's something that we can use freely, that we even adapt to novel use cases. If this is possible, then I tend to try to use open source tooling, because it also is much easier if something pops up to fix something, to get help even from outside the company. You can't fix everything yourself. If it's a black box software tooling, then it's usually very difficult to fix things without talking to the supplier. Might be fine for some use cases, but I found it very difficult, especially for this quick-moving biotech, bioinformatics tooling, assay development, to find somebody who does it in a way that you can actually just employ it without actually checking that it does the right thing.
So that's focusing on whether the tool works by the sound of it. And we talked also about the record keeping and other stuff. Are there other considerations beyond just the raw functionality?
Quality is also a big thing. You need to identify whether tooling is actually fit for purpose. That means you have to have software tests. Ideally, you would like to see documentation for the tooling so that it's not completely unclear what it does. It really depends a lot on what this tool does. So if it's just an R script that somebody wrote, it's relatively easy to check, because you can even come up with a copy of that documented properly and then just use this. In other cases, much more difficult. For standard tools, let's say SAM tools or something like that, to read SAM or BAM files, I think there is documentation, there is code testing, there is hundreds, if not thousands of people using this. There's a lot of trust in the tooling that it does what it should do, and that you can also then to say this is probably valid and okay code to use. Other cases, if it's a one-man show project from somebody who doesn't have any documentation up, no tests, no user base, it's getting much more tricky. And that's also something that we briefly touched upon when we had the kickoff with the regulatory special interest group. One of our auditors, in-house auditors, was also joining, and he also commented on that it's a lot about the trust that you have into this software tooling. And trust comes with tests, trust comes with test data and documentation, and also user base. Because if you have thousands of people using this, it's very likely that it's either stable, or if there is a bug that is there, then it's going to be found and then hopefully also fixed.
So effectively, you have to be prepared. If an auditor asks you, why did you choose this software and how do you trust that it works, you need to be able to give them an answer.
Yes, exactly. You have to have documentation for that. You have to have some document where you outline, this is what we did to check. This is why we trust this. This is why we think this is fit for purpose.
You're in academia and in QBiC, like how much of a change is this? What percentage of overhead does this requirement add?
30% overhead, at least. It depends heavily on what you do in the end. It depends also in what settings you do analysis, because also analysis is defined scopes of analysis. So for example, if you do analysis on a very exploratory level, then because the data is not going to be used for anything where you do decision making in the end, then it's fine to do it a little bit less strict, which means your overhead will be lower. In other cases where you have way more stringent requirements to be followed, so for example, if it's something that you use for decision making in a clinical trial, do you actually continue this? Does it make sense? Then it's getting much more difficult, because then you actually have to make sure that what you do is really properly 100% documented and also fit for this specific decision making purpose. So that's a key difference, I would say. It really depends. There's no general answer to that, I think.
It's pretty significant. Before we started this podcast, we were chatting a little bit and you were talking about some work you'd been doing with biomarker analysis. Is that right? Can you tell us a little bit about your experience there?
Yes, it's not that different to what other people are also doing in like academic institutions or bioinformatics core units, I would say. What we do is we get a lot of patient data from clinical trials. For example, let's say standard types like RNA-seq, but also more exotic ones like mutational calling, some specific assays that you actually get readouts from, and then you have to process that data using. But you can actually use Nextflow pipelines in-house and of course, whatever you want, as long as it's fit for purpose. And then the outputs of that, you then also have to do downstream analysis. Do I see some genes being differentially expressed between my treatment placebo group? Do I see something over time, for example, so longitudinal analysis, so that you see, okay, a patient got the first dose and I see a certain group of genes which we hypothesize about going up in regulation or something. This type of analysis is usually something that you do in R or Python or something like that, in a pipeline setting anymore. That also has to be reviewed and quality controlled. You also have to check that the code that somebody wrote actually does the right thing, that it's properly documented, that it doesn't do nasty hacks, for example, with the data, so that the code is really also something that you can really reproduce in a year, 2 years without having any issues or even longer time frame. It also is stored away, that it's versioned, that you can actually make sure that everything actually ends up in a long-term story.
So, I keep pointing back to previous podcast episodes, but the last one, 43, but was about data studios, which is this new feature we're developing at Secure. And this is exactly the thing we were talking about there. I think I was saying, like, there's no point in having a super reproducible analysis pipeline in Nextflow if you then go off and do something totally undocumented and non-reproducible for the final stage of the analysis. I think that's one of the more difficult parts of what we do with reproducible bioinformatics.
It's also more difficult in a sense that for a pipeline, it's usually very much defined what it does in the end. I mean, it's like a fixed input, fixed output, more or less. If you point it to a certain code revision, you just use that code tag and then it does exactly what you want. After that, freedom starts to some extent. You can actually do a little bit more in various directions, which is also, I think, fine to some extent, because you also need to explore, you also need to make up your mind what you can actually see. Are there some trends from a different angle now? This is a bit different than what you actually do with the pipeline, but I fully agree. It should become the point where you actually lose all of this reproducibility and trackability and of your analysis, because then what's the point in doing the pipeline reproducible if you just give up on anything after that?
Exactly, yeah. Tell us a little bit about your thoughts with regulatory work and how we started off this NF Core special interest group.
So we had internal discussions on how to move forward with these regulatory discussions around the pharma context. But from a pharma perspective, there's a little bit of a push towards making also the pipeline part a little bit more open in terms of that you can also run pipelines in a validated setting. So that is also some requests, for example, for future audits, that potentially FDA and EMA as regulatory bodies in the US versus the European Union are actually also requesting this pipeline. For example, FDA also has this biocompute project nowadays, but they also run pipelines and try to find out whether they are really reproducible. There's some push from these authorities, I think, in that direction.
Yeah, Boston earlier this year, the Nextflow Summit in Boston, there was a talk by Jonathan, who's one of the original people working on biocompute objects. And he talked a little bit about his involvement with the FDA. The BCO files are now officially recognized, I think, by the US Food and Drug Association. We also have RO crates in Europe, which the two are very similar. It's a big JSON object describing all the outputs of a pipeline and what ran, so that you can look at a standardized file format. It doesn't necessarily matter what the pipeline was or what language the pipeline was written in, but you have a standardized JSON file that the auditors can look at and understand what ran and what was produced.
Yeah, I think exactly these type of developments are nowadays starting and also have been started like a year ago or 2 years ago maybe. But they're still early there. For example, a lot of the pipelines that people are using are usually in-house pipelines, are usually validated in an in-house setting. You also have to make sure that all the documentation fits with your own quality framework that you have within your company. So that might be different between companies as well, to some extent at least. Although it's usually aligned with what the authorities want. But what our thought was with this push towards doing this with NF Core was that there is a large overlap between companies, I think, doing them more or less the same thing. We've also seen people from Novo Nordisk in the regulatory kickoff meeting. We've also seen people having interest from other pharma companies, other providers who are also having interest in this kind of topic, because they're probably seeing the same issues that we have as well. You have to make sure that documentation is up to date. You have to make sure that there are tests for your pipe. You have to make sure that there are certain guidelines that are adhered to. You have to prove that. You have to also document it in-house to some extent. But if we come up with good standards, good ideas on how to do this within NF Core, how to employ the NF Core standards that are already there, it's not like we don't have anything. It's a huge framework around that. NF10, I mean, it's developing quite substantially over the last couple of years. It's really amazing what you can actually do now. In my opinion, it's just that you have to point people to that you have to make sure that, for example, if there are new updates coming for a new pipeline, that they are reviewed properly, that there is some tests done, that there is some documentation updates. And then I think we can push it in a direction that helps everybody.
That's my feeling with much of this regulatory stuff. It's not really necessarily anything novel. It's already just best practices that everyone in the research environment should be using, whether you're in academia or pharma or anywhere. Pull request reviews and tests and documentation are a good thing. I don't think anyone disagrees with that. I guess the point of the regulatory work is just to ensure that is in place.
Exactly. And I think that's the point. We would like to look into NF Core, what's already there, what is already available. Are there ways to also more openly, more actively also promote this a little bit? Because one of the feedbacks we got from our quality colleagues was it wasn't so easy to find things like that. For example, for somebody who's not involved in NF Core at all, and usually an auditor would not be involved in NF Core, they would like to find this very easily. And our idea was, can we collect this feedback? Can we collect all of the interesting bits that we need to promote to these people with an audit background, promote to authorities as well, that this is already done, that we adhere certain standards, that there's guidelines out there that people have to follow, and then put this on a summary page, something like an extra label on pipelines that adhere to this very strictly. Things like that could be discussed, could be brought up and formed in the community, and then can help people doing this qualification of these pipelines later on much better than it's currently the case.
At the moment, you have to say to the auditor, we've got a 3-day training course on Nextflow you can drive. It's maybe not going to fly.
Maybe not.
I want to just quickly take a step back as well. The idea of this special interest group then is a way to bring people together working in a specific field or with a specific interest, but across multiple NF Core pipelines. So the discussion isn't just siloed per pipeline. And then try and foster collaboration of other things. The community collaborates in many ways. It's not just writing code. It's not just bug fixing a specific pipeline. This is such a good example of where we can collaborate in a secondary way. The regulatory special interest group is, I think, is the second one that's been set up. If anyone is interested, you go to the NF Core website, hit community, and there's a page for special interest groups here. So we've got 4 of them live as of today, and regulatory is there with a little description and list of key people and list of key pipelines and stuff. Without going into loads of detail, maybe Alex, tell us a little bit about how that started and how it's been received so far.
And the kickoff was in early July. Lots of people actually joined in with different backgrounds. We had people from pharma context, we had people from academic institutions, core units, SciLife had people, QBiC had people, for example, to just mention where we came from. But there's also people just from the community who don't have an affiliation at the moment. There's also care providers who joined in. And we even had people purely out of interest. And I think the kickoff went very good. It was a lot of collection of ideas, a lot of collection of what people see as in scope of this regulatory special interest group. The difficulty now, I think, is to really come up with a more stringent way of looking at these things, to collect all of these ideas. We already started Google Docs that people could contribute to, which already has quite some substantial cross-links also and cross-references to a lot of different initiatives where people already started collecting topics around regulatory topics. Now we actually have to go through this and maybe also sort out things in different subgroups so that we can focus on these distinguishedly in separate subgroups. I think there is a strong community of people from a pharma background who would like to more focus on more authority-related points. However, we also have people with a clinical diagnostics background. I think there is an overlap between what we do in both settings, but we need to identify first, where is this overlap exactly? How large it is really? Can we take an influence? Can we also come up with standards that would apply for both? And in other cases, are there some standards that we would like to specifically highlight? Okay, if you want to use this pipeline for anything that goes past this point, then you would have to obey these rules, this rules. Make sure that you follow these kind of guidelines. It's a bit early to talk about that because I also have to digest it. 20-pager nowadays already, and I don't have had the chance to actually go through it entirely. But I think there's a lot of people who already have some background in this and are willing to contribute, and that's already a good success, I guess.
I think with the attendance for that meeting and the enthusiasm that people hit with it really shows that this is a key area that many people are interested in. You never really know until you actually try and organize a kickoff like this. But I think just the number of people commenting on LinkedIn about it and stuff as well, I think it's a real hot area. And what I love about it as well, like all of NF Core, it's very grassroots. Like the people in that meeting, the people driving this are the people who are actually doing this work, and it's in everyone's best interests, and hopefully just it's a win-win situation for everybody.
Yeah, just maybe as a comment on from our end, my hope personally would be that if we come up with good standards based on NF Core guidelines and maybe also add some additional guidelines for these specific settings that we would like to target for, that authorities like FDA, EMA will just pick this up together with us, maybe even just say, okay, it looks very good. We can actually have this also as part of the recommendations on how to do these things properly. Because at the moment, there is not that much around. Other people from other companies tell us more or less the same. They have their own standards, they have their own developments to some extent, and they try to make sure that they follow exactly what is out there. But If it's not covered yet, then I think it's a good time now to collect across the community how to do, for example, pipelines properly so that you can use them in such trial settings, and then also bring that up with authorities. And our intent also was to actually invite people from authorities to also join these regulatory special interest group calls at some point, and maybe also do a presentation and bring their perspective in.
Exactly, because it's in their interest as well. I imagine
Yes.
If you're an auditor, you don't want to have to figure out and understand 10 different systems. If everyone's just using the same pipeline, it's going to save you a lot of time and effort.
Yes.
So you've touched how you use open source software, but obviously you're a keen advocate of open source yourself, and you've written NF core pipelines from scratch whilst being at BI. I've forgotten the name of the pipeline now.
NanoString pipeline, for example, is one of the contributions we made.
And so you've written from scratch there and pushed to NF core and made that open for the community, which I think is fantastic. One of the things I think people maybe outside of pharma often view pharma with a bisque slant that it's very commercial. There's lots of things about privacy, data protection obviously is important, but also down to the code and intellectual property and everything. From your point of view inside such a company, obviously you're using and contributing a lot of open source work, which might be surprising to some. Is that a cultural difference, or is it a wrong assumption for people to make?
I think it is something that is about to change, or it was changing the last couple of years. A lot of people that I've met at different conferences with also pharma background are more or less telling me the same stories now. It's impossible these days to actually do analysis on any of the novel data types that you've seen across clinical trials, clinical research, without using open source tooling. You can't just reinvent the wheel if there's a proper tooling out there with open source. And in the end, it boils down to, is it something that you just use, or is it really something that you also have to understand? In my opinion, you also have to understand these tools, because in the end, you also have to prove that you understand what you're doing there. If something is broken, you have to fix it. And if you can fix it in an open source way, in my personal opinion, and I think a lot of people think very much the same also in the pharma context nowadays, it's much better to fix it upstream in the open source community, in the actual project that works with these tools. Let it be challenged there as well, critically be challenged. I know who I'm talking to here. Your pull request reviews are famous for me as well. For people who don't know, I also have a special title in NF Core, the Merge Cowboy, because I was keen of merging pull requests a bit earlier than most people maybe. Phil was always one person who was really very strict about pull request reviews and gave a lot of feedback, which helps in many cases also to improve it. That's also something you have to be aware of.
I think it's going back a bit now. I do fewer pull requests than I used to. I think it's a somewhat dubious reputation to have, as I'll know Phil's going to drop 200 comments on my pull request. But I think it's, especially in the early days of NF Core, it was important. It helps with that standardization a lot.
Yeah, it really helps. And I think that's also something changing with pharmas now as well. I don't know about how it was 15 years ago, 20 years ago. I wasn't working there, but my personal experience nowadays is we have multiple people who are also in multiple open source communities, multiple open source projects who are actively contributing. Obviously, there has to be a clear focus. It's not like I can contribute whatever I want. I can't work on ancient DNA analysis pipelines anymore. But if there's a good justification, it's usually possible for people.
Unfortunately, mammoths don't need very many drugs.
They won't help anymore, I guess.
I'm personally super excited about this topic. It's hugely important to good science, to good healthcare, good development. I feel like this is a pivotal moment for NF Core a little bit, and hopefully as part of a larger bioinformatics picture of coming together, collaborating, helping one another. And I'm really excited by the enthusiasm that we've seen in the community so far, and very keen to see what comes out in the months to come. The next regulatory meeting is coming up pretty soon, right?
I think it was set up for September 9th.
Still plenty of time for people to join and contribute to that. And then after that, the next thing will be you're giving a talk at the summit in October. So there's two fairly big milestones coming up for that special interest group. Is there anything else that would be useful for people to know who are listening, who are interested in this, to find out more information or get involved?
If you want to get involved, please just join the Slack channel. Please just join the group there. We'll announce every meeting that we will have in the future on the NF Core webpage. My focus is clearly on pharma, obviously, because that's what I do. But there will be probably also subgroups working with a different angle on these topics and collecting information around this from a different angle. We will discuss this, and it's really early days still. We just had to kick off. Please just get involved.
And the Slack channel is called #regulatory, so that's the best place. And all of the meetings are being recorded or live streamed and going to YouTube. So I think we've got a playlist called NFCore Regulatory Special Interest Group. So there's a playlist which will be maintained. At the moment, there's just the one talk in it, which is the kickoff meeting. So even if you missed that kickoff, you can still go back and catch up. Thanks so much for joining me today. It's been an absolute pleasure, as always, chatting to you. Looking forward to seeing you in person in a few weeks.
Yes.
And anyone listening, just final plug, remember that early bird 20% discount is going to run out in just a couple of weeks. So get your skates on, get that ticket, and hopefully meet many of you listening in person in a few weeks as well.
Join the full week, not just the summit. The hackathon is also super fun to join. I've really missed the last ones, and this time I really look forward to going.
This is not always super clear to people. People see the word hackathon and they think, oh, it's just going to be super nerdy coding. But there's a big chunk of people who really spend most of their time just chatting or planning. You see all kinds of outputs from a hackathon, which are not just pull requests. It's bigger than that. And this is a great example of that. Fantastic. Thanks very much, Alex. And
thank you.
See everyone soon. Cheers. Thank you.